First of all, I would like to thank the organizers for organizing such a nice conference. I quite enjoyed a lot the talk today. Starting from Lauren to the last talk, I think they've been brilliant. I think following up on Lauren's talk on open science and so on, I thought that I might as well share with you my journey into this project, which is actually not the main project that I work on for my postdoc, but it was very much driven, I think, by open science, or at least some part of open science. So I'm going to share the journey with you. So how I got involved into this project. So on the 30th of September, I read this tweet by Jesse Mayer. So he's a new PI in the States, and then he was asking whether someone was interested in doing the revisions for a paper that he submitted, he was. So someone who was fluent in R and proteomics would be able to make some of these revisions. He also posted on the thread a link to his GitHub repo where the project was described, or was he basically put all the script into that repo. So I read that tweet the day before I was taking a flight. And being in the UK, that meant that I had to self-quarantine. So I will have some time, and I did my PhD in proteomics, and actually for my postdoc, I work as well in proteomics, and I'm fluent in R. So I thought that I went into the repo, and also Jesse posted a preprint on bioRxiv. That's why this was very much driven by open science. So I read the paper. I thought it was a cool project. So we shared some ideas, and then we decided to give it a go. So I forked Jesse's repo, and then a couple of weeks later, I put his project into a package, and now the paper has been accepted into the Journal of Proteome Research. So the project that we work on, we call it PCORA, which stands for Peptide Correlation Analysis. So I'm gonna explain you now what is this project about. So basically, from the talks that we hear today, we know that in shortcut proteomics, peptides are produced from proteins by proteases like trypsin, as you can see here. So then after trypsin treatment, this generates some peptides that you can see here. So then the protein quantities are inferred from the group peptide quantities of the experiment. So in this example here, you can see that the level of this peptide, this protein, is increased after this treatment, and you can see as well that the individual peptide of that proteins increase. So the three peptide of that protein follow the same direction. So the three peptide increase upon treatment. But you can have also the scenario where the level of a protein doesn't change after the treatment, as you can see here, but some of those peptides might have some post-translational modification. So, for example, this green peptide here that gets phosphorylated. So then the level of the individual peptide for that protein disagree. So, for example, this green peptide, the total level of that green peptide in that protein is lower, right? So, in these scenarios where you might have discordant peptide, like the green one, they represent a problem. So multiple strategies have been focusing on the detection and removal of those discordant peptides. But the concept of gaining proteoform information from those discordant peptides is greatly underexplored in the proteomics field. So this is what the PCORA algorithm aims to solve. And so this is how the workflow of PCORA is. So there is an initial global scaling and center of peptides per replicate. And this is something that we actually do normally in a proteomics workflow. So there is another step where each peptide is scaled to the center of the control condition, as you can see here. And then the Pecora algorithm fits a linear model for each individual, for each unique peptide in the protein. And this model includes an interaction between the peptide groups and the treatment, and it performs an ANOVA test. So the ANOVA test calculates a p-value for testing whether each peptide agrees with all the other peptides in the protein. And then the p-values for all the peptides are recorded, and there is a multiple hypothesis testing correction applied on a per protein basis. So there's a last step where there's a scaling step that improves the statistical power of the p-value for interaction. So to develop this method, Jesse used a data set deposited in a prior repository. So again, like this was open data that the community can access. And the data set that Jesse used was a study where the authors aimed to reveal changes in mouse primary microglia upon ethanol exposure. So that's why you can see that there's a control condition. There's a ethanol treatment. I think this was 50 millimolar. And also there's another condition of LPS treatment. So the authors wanted to see whether there was any changes based on, mediated by inflammation. So we used those data and then analyzed it with Pecora, and then we found that the most significant adjusted p-value correspond to a peptide present in the protein kinase type 1 alpha regulatory subunit. So as you can see here. And then specifically it was on a peptide that contained an oxidated methionine on residue 331. So this peptide was also detected without the oxidation, but only the oxidated peptide was significantly increased due to the LPS stimulation, as you can see here. So pKa is a kinase involved in cell signaling, and it is thought to be regulated by oxidative stress. So the methionine 331 is on the kinase surface, as you can see, and is near the CMP binding. So this oxidation could regulate the CMP binding or other protein-protein interaction of the protein. But importantly, this direct observation of this regulated PTM site was uncovered by Pecora and would have been missed using a standard protein quantity summary that aggregate peptide quantities. So this represents an example on how Pecora can detect regulated PTM changes. So we wanted to show whether Pecora can detect differential proteoform changes in other dataset types. And so we performed Pecora analysis on a recent published dataset containing protein signatures of patients with COVID-19-driven acute respiratory distress syndrome, or patients that didn't have COVID-mediated acute respiratory distress syndrome. So we used this data set. And so a prevalent characteristic of COVID-19 severity is high prothrombin time. So this is the time that takes prothrombin, so this protein involved in coagulation, because basically it interacts with fibrogen. So it's the time that takes the inactive prothrombin to be cleaved into the active form, so active thrombin represented here. So basically what we found using the Pecora algorithm was that a peptide on the N-terminal, on the inactive prothrombin form, was significantly higher in COVID-19 patients. So this differential regulation of thrombin isoform in COVID-19 patients may partially explain their elevated risk of thrombosis. So to understand the biology of different proteoforms associated with COVID-19, we perform a gene ontology enrichment analysis on the differential proteoforms detected with Pecora. So this analysis revealed that the pathways with different proteoforms were related to proteolytic processing cascade, including proteins from the complement activation, as you can see here in green, and also proteins involved in blood coagulation, as you see here in red. So this is an example on how PECORA can detect different proteoforms. So this was the main aim of PECORA, actually, so to discover differentially regulated proteoforms. But we wonder if PECORA could also improve protein level quantitation if used as a pre-filter for discordant peptide. So this is something that other tools have been developed and used for, such as the MSTAT method. So if you work in proteomics, you might be familiar with this method, and it's also available as an R package. And so one of the data sets that the authors of this project used was this data set that was acquired by a data-dependent acquisition mode, and is is available. So we use that data set to benchmark how our PECORA pre-filtering algorithm could improve protein level. And then we compare the MSTAT approach and our PECORA approach. And so what we saw was that the quantitative quality produced by both feature selection was pretty similar, as you can see in the scatter plot here. And we also found that the PECORA prefiltration reduced the number of false positive, as you can see here. So similarly to the MSTAT feature selection approach. And so I'm going to show you an example here on how excluding discordant peptide can indeed eliminate some false positive. So this is actually from the MSTAT's original paper, and so what the authors wanted to highlight in this example here was that they were doing protein level quantitation, and then they focused on this protein. So they had 4 conditions for that experiment, and then what they saw is that if they include all the peptides that they call features, they saw that this protein was significantly changing between condition 1 and condition 2. So it's represented in this table here. So the log 2-fold change between those 2 conditions is significant. But however, what they saw is that when they applied that feature selection, so when they were removing some of those outliers, the log 2-fold change was not significant anymore. So they were removing those false positive. So what we saw on using the PECORA algorithm was that we were indeed detecting those discordant peptides, and indeed when we remove those outliers, we were reducing the number of false positives. So with this, I wanted to, I want to highlight how we developed a method to get quantitative proteoform information from bottom-up proteomic experiments. We also saw that the method can perform on data sets processed with different tools. So actually some of the data that we analyzed came from data sets processed with MaxQuant approaches, Skyline, Progenesis. And also using PECORA as a pre-filter step can improve the number of false positive, so can reduce the number of false positive detection when we were doing protein level quantities. So we put this algorithm as an R package. So if you go to Jesse's GitHub repo, you can have a look at the vignette and also see where are the functions available in PECORA. And so with this, I want to acknowledge, first of all, Jessie for, making this project available to the community and for, allowing me to chip in to this cool project. Also my boss for, giving me the opportunity to work on this project that is actually not the main project that I have with him in the lab. The RSG consortium for giving me the opportunity to present the work here, and also the MRC who ultimately pays my salary. So with this, I would like to thank you for your attention. I'm happy to take questions. And also, I will be presenting this work in the Bioconductor 2020 in a couple of days. So, I can take questions there as well. So thank you very much.
Thank you a lot. Nice. See you then at the Eurobioconductor. A very, very nice talk and a nice story also at the beginning. How it's amazing how some research project starts from just a tweet, let's say. I see here Chong has a question. Have you compared your FDR before and after any validation algorithm? For instance, percolator.
I don't understand. I mean, I don't get what he means by before and after. Before and after, like which step?
Maybe if Chong, if you could rephrase your question, I can maybe ask another question. Meanwhile, I was wondering. So, I think there was one part that I maybe missed, but so you thanks to pCoRe, you can identify discordant peptides, right? And my question was, so the idea is once you identify those, you discard them, or?
No. So the idea is not to discard them, but actually look at them to see, But then the thing is, like, you want to do, protein quantification, so like, grouping peptide, then it might be, you might benefit from discarding them. But the idea is not to discard them, but actually look at them.
Okay, right. Cool. Because
you have to stay in the, peptide level. So like when you are doing that, you are actually looking in peptide. You're actually comparing peptide.
Okay. And do you think, because I think it's very interesting identifying those discordant peptides, do you think maybe at some moment we could, you told you did, for instance, some ANOVA, but including them in some way so modeling them when doing differential expression analysis or so, something like that?
Yeah. So, so this is how we develop it. And it was pretty, so it's pretty recent as well. But, I also have some ideas things that we could do to improve the algorithm. So, for example, like, we didn't include what is the, like the mascot quality of those peptides and stuff like that. So, I think we could improve the model. The model at the moment is just following a standard, like a linear regression. Okay. But like we can do so many things that, like, peptide quality and
very cool. Like okay, I see Chong rephrase this question. He means before and after using any validation rescoring application. I think peptide prophet and percolator are both used to improve FDR validation. And then
so that's a very good very good question. We didn't do it. I mean, we do FDR correction and the last step, but I haven't looked into percolator, but there is something that we could certainly do to improve the model.
Okay, and I think he also asks, do you mean you prefer the peptide level validation instead of aggregate them into protein level?
I think it's depending on what you are interested in. Normally in proteomics is pretty much everything is based on protein groups, right? But I think it depends on your question. But I think, if you are, for example, interested in different protein isoforms, maybe you can benefit from this. I think it depends on the question.
I see Jessie is responding to Chong's question. PerCoA is after the identification process. It's agnostic to whether peptide prophet or percolator was used. Okay, thanks for this additional comment.
it's Jesse who originally wrote the algorithm. So
okay, great. Thanks a lot. Well, thank you very much again for this talk, for this actually very last talk of the RSG Belgium online symposium.